Kevin Davies, you are an editor, author, and public speaker, and you are the executive editor of the CRISPR Journal. And today we're here to talk about your fourth book, Editing Humanity, the CRISPR Revolution and the New Era of Genome Editing. Kevin, thank you so much for being here.
Thank you, Alan. My pleasure.
So, Kevin, when I'm reading your book, I actually feel like I'm reading a science fiction book. I still, every time you say something, I had to go to YouTube to make sure it's real. You know, it's just, is this really happening? Is this real possible? So you have blown me away. And I guess many of my listeners and readers of your book will feel the same thing because science is going faster than fiction.
Yeah.
I wonder if you could start by telling us a little bit about your background. How do you get into this field of expertise?
Sure. I'm a geneticist by training, and the original career plan was by this time I would be, I don't know, a professor somewhere at some famous hallowed university. But that didn't work out. My research skills were much less than I thought in my younger days. And so after moving to Boston in the late 1980s, I realized I had to find another way to make a living in science, which was the only area I'd learned growing up in the UK. And I found a way to do that by joining the editorial staff of a famous journal called Nature, and got a couple of lucky breaks while I was there to launch some new journals, one new journal in the genetics space. And I've stayed in science publishing all along. And when a big story in science, and especially in genetics, the field I love the most, comes along, I usually feel motivated to try to weave a narrative tale around that story because I feel that it may have broader medical and societal significance. So I've written books about the race for the breast cancer gene. That was in the mid-1990s. Featuring the wonderful geneticist Mary Claire King. Then I wrote a book about the race to complete the Human Genome Project, which was quite spicy at times. And then I wrote a book called The $1,000 Genome, which was about advances in consumer genetics, the rise of companies like 23andMe, and massive progress in DNA sequencing. And then a few years ago, I suddenly got switched on to CRISPR and the ability to not just sequence or read DNA sequences, but to actually edit or correct them. And that for me that's kind of a holy grail for a geneticist. And I just felt compelled to write a book on the subject.
Okay, can you tell us what is the Human Genome Project, just for people like me who don't know?
Yeah, sure. That was an international collaborative project to once and for all sequence the DNA of a human being. So our DNA is bundled into chromosomes. We have 23 pairs of chromosomes in virtually all of our 10 trillion cells. And the Genome Project was an effort to decode every single letter of the genetic alphabet from the beginning of chromosome 1 to the very end of chromosome Y, the male sex chromosome. So we would have a roadmap to find genes that govern our development, our behavior, and that go wrong in diseases, from straightforward genetic diseases like cystic fibrosis or muscular dystrophy to much more complex genetic conditions, heart disease, schizophrenia, mental illness of many different forms, where there's likely to be an interplay between different genes and various environmental triggers. If we don't have a roadmap, if we don't have the full sequence of our DNA in front of us or on our computer screen, we're going to be handicapped. So that was an effort to lay that foundation once and for all. It was completed in its first draft in 2000. There was a famous White House ceremony hosted by President Bill Clinton. And the world of genetics and many other fields besides have flourished as a result of having that genetic roadmap.
Okay, and then according to your book number 3, I believe that the initial cost to read this genome was about $1 billion. And then when you wrote that book, it was down to $1,000. Can you tell us more or less what's the cost nowadays?
Well, yeah, there are companies now claiming that you could probably sequence the genome for, in terms of raw costs, raw materials, for as little as a few $100s. So yeah, the $1,000 genome was an extraordinary, you spoke of science fiction in your introduction. I heard that term for the first time in 2002, and I thought the speaker, I thought he was high on something. I mean, I just thought that was ludicrous that we would go from the Human Genome Project, which literally costs $1 or $2 billion to assemble over a decade, to now talk about decoding somebody's personal genome for $1,000. I thought that was madness. But new technologies were rapidly being invented, and those have taken root. So now, today, all the big medical centers use new sequencing technologies from companies like Illumina and PacBio and Oxford Nanopore and others, to the point that we can, sequencing a genome now is pretty easy. Making sense of it, interpreting it, that can be a bit more challenging.
And okay, so for a regular person like myself, what would be the advantage of reading my whole genome sequence?
Well, whether you, as a just walking around today with no particular health problems, whether you need to or want to read your own human sequence, your DNA sequence, is a matter of personal choice. No one is saying you have to or you necessarily should, but the ability is there, and some people have learned many very important things from looking at their full genome sequence or looking at the edited highlights that you can get from a company like 23andMe or Ancestry.com. You may learn something very important in the medical realm about genes that you carry, or potentially diseases that you are at risk for, that you may have had a vague family history but weren't acutely aware of what the problems might be. And there could be very specific drugs or other medical interventions that that could potentially be life-saving. And there have been many stories over the last decade of people who perhaps were adopted, who used DNA information to find relatives or learn things about their families, sometimes for good, sometimes not. But what companies like 23andMe have done is they've allowed us as individuals to take more responsibility. We should, I think, ultimately be able to have access to this information if we want. It shouldn't come with the paternalistic pat of a GP or a medical doctor. This is fundamentally your information, and you should have access to it.
Okay, so now we come to this book, and you talk about CRISPR and the ability to edit this sequence. What exactly does that mean? I mean, is someone sitting in a computer changing things around, or just mixing liquids in a test tube? What is exactly editing your genome?
I'll start with the headline, and then we can maybe work backwards. CRISPR does give us, in principle, the ability to look at the DNA of a patient with cancer or with sickle cell disease or a growing list of genetic diseases, and go in and potentially make a fix to the DNA code as small as what's changing one tiny letter, like correcting a one-letter typo in a book, to potentially correct the effects of that genetic disease. The most exciting illustration of that to date was published late last year, late in 2020, in the New England Journal of Medicine, as investigators reported on the treatment of a woman named Victoria Gray, a sickle cell patient from Mississippi, who had spent her life suffering from very painful episodes and crises caused by her disease. These physicians and investigators at CRISPR Therapeutics and Vertex Pharmaceuticals have been able to go in, take out some of her stem cells, make changes that can switch on a gene that is otherwise silent to essentially correct the effects of a mutation in a related gene. And the results have been nothing short of spectacular. She is essentially cured. She's reported no adverse events, no painful crises in over a year since she was treated. So that, although the numbers are very, very small, that shows the potential clinical promise of this new gene editing technology called CRISPR. And the scientific community and the broader community have recognized this because, as many of your viewers will know, the two female scientists who developed this technology, or certainly received the lion's share of the credit for developing the technology, won the Nobel Prize for Chemistry last October, Jennifer Doudna and Emmanuel Charpentier.
Wow. Okay. So my head is about to explode. So scientists took a part of I don't know, stem cell. I think I understood. Then they play with it and then put it back. Is that what you said?
Yeah. So let's just say a little bit about what CRISPR is. CRISPR, when it was first discovered by and being studied 20, 30 years ago by a very small number of scientists, it had nothing to do with treating genetic disease. This was a phenomenon, a system that was discovered in bacteria and other microorganisms. And over the course of a decade or 15 years, scientists began to piece together that bacteria contain a motif in their DNA. It's now nicknamed CRISPR. That provides, it's sort of their antiviral missile defense system. So in this CRISPR section or compartment of the bacterial DNA, bacteria are able to store away, file away segments, snippets of the DNA of viruses that are otherwise trying to kill them. And this is used as a sort of defense mechanism, such that if later on the bacteria should encounter the same virus, they have a record of past encounters with this virus. And this DNA sequence can be activated, armed or coupled with an enzyme that is able to cut incoming DNA. And the queue, the sequence that was previously stored, provides in the bacterial genome, provides the molecular target so that these enzymatic scissors, or the genetic scissors, as the Nobel Prize Committee called them, don't just go cutting indiscriminately. That would not be good for any cell, but are programmed to only cut when they latch onto that specific sequence. In this case, we're talking about a virus. The work of Doudna and Charpentier, building on the work of many other fabulous researchers, showed that we could modify that system to now target, in principle, any DNA of our choosing. It could be plant DNA, it could be animal DNA, it could and has been human DNA. The CRISPR system makes a cut at this pre-specified target sequence, and we can use the cell's own repair processes to then fill in the gap caused by that cut sequence and make a subtle change in the sequence that suits our end. So if we're trying to correct a genetic disease, we can cut near or at the site of the mutation and figure out a way to splice back in the normal human sequence. Since the discovery of CRISPR, researchers, notably David Liu's lab at Harvard and the Broad Institute in the Boston area, and others as well, have developed new gene editing technologies, one in particular called base editing, that can perform even more precise single letter changes. It's chemistry on the genome without even completely cutting the DNA. So it's safer and more precise. And although that's not yet in the clinic treating human patients, I think it's only a matter of time based on some of the early preclinical results that have been reported quite recently. So it's an incredibly exciting time for CRISPR, for the gene editing field, because we now have the potential, and many companies are pursuing this, to treat a whole array of genetic diseases and potentially other diseases. Maybe one day we'll be able to use this to treat infectious diseases, maybe even viral diseases like COVID-19. But there are many other applications as well. Maybe we can engineer safer, healthier forms of crops and livestock so that we can secure the ability to feed the planet as our population continues to grow.
So this sounds like designer medicine or custom-made, yes, drugs or fixing. What are other cures or that are in the works right now. The
Yeah,
scientists have focused their effort into, okay, we
yeah.
have this whole world of potential. Let's focus on this right now.
There's a growing list of diseases that investigators are tackling because thanks to the Human Genome Project, we know at the molecular level, at the DNA level, what the specific typos are in our DNA that give rise to thousands of genetic diseases. And in principle, CRISPR or some of these other gene editing technologies, such as base editing or prime editing, which is another very exciting new technology, can, as long as we know the sequence that has gone wrong, investigators can design the molecular targets to go after those specific sequences and not just land randomly in other parts of our DNA. So much work has gone on in the last 5 years or so to make these signatures as precise and as safe as possible. And now there are clinical trials ongoing to treat hereditary forms of blindness, for example, to look at certain liver diseases. Soon we'll be tackling hemophilia, muscular dystrophy, and I'm sure that before long we'll be thinking about using this for neurological diseases, maybe certain forms of Alzheimer's disease or other things. That doesn't mean that CRISPR or gene editing is going to provide a cure automatically. Treating genetic diseases, treating diseases of the brain is very difficult. Any form of drug intervention can have its safety ramifications and things can crop up. But this is a tremendously exciting time for genomic medicine.
It's just crazy. So you open the book with a story about the CRISPR babies, and there's a Chinese scientist. I don't know if I pronounce his name. Jiankui, I think.
Oh, Jiankui, yes, exactly.
Okay, perfect. So can you tell the audience exactly what he did, and then what are the legalities and the ethical problems that we are seeing in regards to
thank you very much. Yes, I spent a fair bit of time halfway through the book. A major event happened in China that brought in a whole new series of ethical questions about genome editing. And so I spent quite a lot of time in the second half of the book sifting through this story with access to some of the principles involved, which was fascinating. What He Jiankui tried to do was, in contrast to all of the other examples that we've been talking about, where we're treating usually adult patients or children, he was editing a human embryo. So he was trying to make a genetic change in a one-cell human embryo that would then be implanted into the mother, and 9 months later be born as a baby with those edits penetrating every cell in that human being. And in late 2018, a pair of twins were born somewhere in China to a Chinese couple, where this action had been performed against the wishes or knowledge of virtually everybody in the scientific or medical community, who were very much opposed to this on a variety of grounds, certainly the safety, but many people felt that we just had no right as a species necessarily to be playing God, to going in and changing the DNA, not just of an individual or a baby, but the baby's, once the baby's grown up to be of reproductive age, his or her children will all carry or could potentially carry those gene edits into their future generations. So we are really tinkering with the gene pool here. He Jiankui presented his results after a tremendous expose by Antonio Regalado, journalist at MIT Technology Review, who scooped everybody and broke the story that he was trying to engineer CRISPR babies, as the hashtag went. The babies were born. We think they're healthy, but honestly we don't really know, because we don't know their identities, and the Chinese authorities have not revealed any further information. And the scientist who did the work, or who led the team that did the work, He Jiankui, is currently halfway through a prison sentence, a 3-year prison sentence, after the authorities caught up with him and realized the severity of what he had tried to do. The quality of the editing that he did in this one particular gene, the purpose of which was to try to make these babies HIV resistant, was very shoddy. It was very poor. And it was medically unnecessary. There are other medical steps that could have been taken, and indeed were taken, ironically, to ensure that these children did not receive HIV. HIV was not transmitted from their dad. So this was a completely unnecessary experiment. The authorities and scientists around the world have reacted and had time to think about what happened and come to some sort of terms with this, and have issued reports. A big one was issued late last year from the National Academies of Sciences, that basically said there may be some very specific grounds in which we might condone the use of this gene editing technology on human embryos in the future, but that time is not yet now, because there's huge safety issues that still have to be resolved. And even once they are resolved, we then have to ensure that this is not done for any frivolous reasons, but only done in very rare cases where perhaps a couple both have a genetic disease and there's no other medical intervention that can be used to give them a biologically healthy child. In those certain narrow criteria, maybe gene editing is a way forward. But this raised a lot of big questions, moral, ethical, religious questions. And I do try to, we could spend a whole another hour talking about them, but that's much of the theme of the second half of Editing Humanity.
What are the probabilities that the Chinese government is admonishing these scientists in public and having, in fact, secret laboratories continuing further the research of this embryo?
There's no suggestion that the Chinese have any secret laboratories. They came down very hard on this scientist who was put under house arrest for almost a year before he was finally sentenced. But I think you do raise the specter that perhaps there is a prospect that in some parts of the world, somebody may decide to set up a CRISPR clinic to offer these sorts of gene editing services to couples who think they want to engineer or have a treatment for a particular genetic disease, or maybe for some other purposes, which I think the vast majority of the medical and scientific establishment would absolutely resist.
Wow. What are the resurrection of a species? What are the possibilities that we may be able to resurrect? I think I read an article not too long ago about, I forgot what was the animal that was being trying to be resurrected, but is this like some Jurassic Park scenario?
We're getting a little bit more into the science fiction realm here, but not completely. I think you're probably referring to the woolly mammoth. So yes, that's one of the examples that I talk about in one of the latter chapters in the book. And this is obviously work led by George Church, a brilliant geneticist who's been at the forefront of genetics for 3 or more decades, and is a font of exciting, original, and sometimes slightly crazy ideas that usually pan out. And the idea to resurrect, it's not literally resurrecting the woolly mammoth, but it's a sort of a multi-step process where there are so many beautifully preserved specimens of the woolly mammoth in Siberia and other parts of the world, that we can build a complete genetic sequence of this extinct species and then compare it to its nearest living relative, which would be the Asian elephant. And in principle, if we were to sort of intersperse maybe a few dozen woolly mammoth genes onto the backbone of the Asian elephant genome, we might be able to create in the next generation a that looks, perhaps to most intents and purposes, more like a woolly mammoth than an Asian elephant. And this isn't for just silly recreational or commercial purposes. Church's goal, and this has been on 60 Minutes, so it must be true, is to have these creatures repopulate a not Jurassic Park, but he calls it Pleistocene Park in the outer reaches of Siberia, to potentially have these creatures keeping the snowpack packed down, such that we don't, thanks to global warming, see vast amounts of methane leaching out into the atmosphere and just exacerbating the climate change problem. So there is an ecological and environmental reason for pursuing this, even though I think a lot of people, including other experts in the field, are still fairly skeptical.
Well, Kevin, I really don't know yet, after speaking to you for half an hour, whether your book belongs in the science or the science fiction part of the library or the Amazon. Can you tell the listeners one more time the titles of the book and where they can follow you?
Thank you so much. Yes, Editing Humanity, the CRISPR Revolution and the New Era of Genome Editing. It was published in October 2020 by Pegasus Books in the United States, literally one day before the Nobel Prize was awarded to Downer and Charpentier. So thankfully, the publisher has agreed to rush out a paperback version, which will be out in May. And my epilogue will talk about the Nobel Prize and some of the other events that have happened since the book was first published. It's also an audiobook. I had the thrill of narrating the audiobook. Wow. And Kindle and hardback right now.
Well, Kevin, thank you so much for your time.
Thank you, Alan. My pleasure. Thanks for having me on the show.